going to present my talk on decoding crop genetics, modern molecular techniques for sustainable agriculture. So I think day before yesterday we have discussed about the importance of future challenges in agriculture, which is addressing the to feeding the ever increasing population with depleting natural resources. So the challenges I think we all know, and these challenges can be tackled by several methods, through crop breeding, crop genetics, as well as through improved economic practices, as well as post-harvest by reducing post-harvest losses. But we are concerning mainly on crop genetics, and we will discuss about its role and its importance. So why we should focus on crop genetics? So crop genetics provides tools to improve crop yield as well as its quality, and it enables us for rapid development of climate resilience crops, as well as breeding for higher input use efficiency, like higher water use efficiency, high fertilizer use efficiency. So by using optimum dose of fertilizer, we can get maximum yield. Similarly, by producing more crops as well as biofortified crops, we can address the food security as well as global nutritional security. And of course it's all have its impact on economic and social benefits also. So crop genetics is the cornerstone of sustainable agriculture, enabling us to meet the challenges of feeding a growing population while conserving natural resources. Why it is important to conserve natural resources? Because a farming approach that meets the needs of the present without compromising the ability of future generation to meet their own needs is very much required. Suppose if we get the higher productivity from a piece of land, but after 1, 2, or 3 decades the land become non-productive, then it is not a sustainable way, and our future generation will perish. So we should focus on sustainable agriculture. So sustainability in agriculture is about producing more with less, like less water, less land, fewer inputs, as well as preserving our environment and ensuring food as well as nutritional security for future generations. So now coming to the modern molecular techniques for crop improvement. So there are, I have listed some of the important molecular techniques, and we will discuss in detail in next slides. But here to summarize, the advent of next generation sequencing techniques enables us to study the plant genomics by a lot of uh, different ways, like transcriptomic, GBS, genotypic by sequencing, whole genome, whole genome resequencing, as well as the complete genome sequencing. Coming to the CRISPR-Cas9, it is a gene editing tool, and this is recently developed, hardly 10-12 years back it's developed, but it has revolutionized the modern agriculture biotechnology, because it enables us to target the precise gene where we can did our mutation. Okay, we can do our mutation. Now coming to the marker-assisted selection. So marker-assisted selection is a technique which we use molecular markers to go for a breeding program, and it enables us to precisely phenotype the plants. Like some of the visible phenotypes breeders can assess, like plant height or bold seed size or larger seed size, high number of tillers. The breeders can assume. But suppose in case of nutritional parameters or disease resistance, the only after the harvest or only after the means a proper disease pressure, the resistant plant can be assessed. But in certain environmental conditions, disease may be skipped. But in that situation, the molecular marker linked to disease resistance is very much required. By using these molecular markers, we can identify the resistant plants even in early stage and remove the negative plants or non-positive plants that don't have the desired gene. So, and the market selection is quite popular in our country also. There are a lot of varieties in rice, wheat, as well as recently even in mustard, market selected varieties has been developed. But now in this market selection, a new technique has emerged called genomic selection. What is genomic selection? It enables us to predict the breeding values of entire present germplasm based on their molecular data. We don't need to meticulously phenotype the entire germ class for getting the for identifying the desired parent for breeding program. So this is very important technique. Now coming to the RNA sequencing and gene expression analysis, so it is a part of next generation sequencing technique we will discuss when we discuss the NGS. And proteomics and metabolomics is also now becoming a very important technique, because we all know about the central dogma of life, like DNA is transcribed into RNA and then RNA is translated into protein. But in these steps, a lot of steps are there where the gene regulation occurred. So suppose a particular, even in last lecture I have discussed this, a particular gene has expressed more, but due to the post transcriptional modifications, that gene may not be responsible for the particular trait. So in that case, proteomic metabolomics enable us to complete the linkage, and from gene to phenotype, there is a complete linkage is there. Like which gene is transcriptome actually enables us to identify the responsible gene which are highly expressed for a certain environmental inputs or stimuli, and that these are messenger RNA transcribed to a particular protein that we will study with the help of proteomics, and that protein actually enables to change a metabolic pathway, and through metabolics we can assess that change. So we have the complete knowledge about the phenotypic expression of a plant for a certain trait by using these omic techniques. So here we have to isolate our DNA from a particular tissue because from a single cell, the detection technique is not that much efficient. But now we can detect even a single cell's genomes through sequencing. So single cell sequencing technique is now becoming new frontiers in our understanding for crop genetics and genomics. Coming to the epigenetics and crop adaptation, so plants actually, what is epigenetics? Epigenetics is the changes in the phenotype without changing the DNA sequences. This happens mainly on the DNA methylation as well as acetylation of the histone protein and other chromosomal associated proteins, and that proteins enables the expression or suppression of a gene without changing the DNA sequences. So from traditional sequencing, we cannot have the information, but through epigenetics, we also have the information about the epigenetic change, and this epigenetic change is highly influenced by the environment, because genetic information is same, the DNA sequence is same, but the acetylation of the histone protein or methylation of the histone protein actually shut down or overexpress a particular gene. So this is due to the environmental changes. So this kind of technique actually help us to complete the picture. And of course, speed reading, I think today one lecture is also there on this topic. So coming to the next generation sequencing, earlier the two methods have been simultaneously developed, Sanger sequencing as well as the maximum Gilbert sequencing. And these two techniques were used to means the foundational technique in terms of DNA sequencing. But during course of time in Human Genome Project, the Sanger sequencing become the priority of the scientists, and due to the lot of funding and many people involved, many labs involved in the Sanger sequencing, so this sequencing has further improved, and it has become groundbreaking technique, and the first of the genomes of any organism was sequenced using the Sanger sequencing techniques. But it is very costly because its techniques have enabled us to means longer read lengths are there, up to 500 or 700 base pair we can sequence in a single run. But the sequencing we have to break down the genome into smaller fragments and the sequence them and then realign them to assemble a complete genome. So it is a very costly technique, and also very time-consuming technique. So around 2004-5, the second generation sequencing techniques has came, and they are called next generation sequencing techniques. But these techniques are like pyro sequencing, sequencing by synthesis, sequencing by ligation, and ion torrent. All these techniques has a capability to use nano volume for their PCR reaction, and very small amount of DNA is required for getting the detection, because detection techniques now become very sensitive. So small amount of DNA can be suppose a few nucleotides added that give the signal, that signal can be detected And but the problem is that these sequencing techniques resulted in shorter read lengths. So now coming to the third generation sequencing techniques, these sequencing techniques enable us to getting the longer DNA reads. And so in this third generation sequencing techniques, we can get the DNA sequence up to several kbs, and the assembling of genome become very easy. But the problem is that the third generation sequencing techniques actually have means the accuracy is not as per as the second generation sequencing technique. So nowadays we are using both these techniques for any genome assembly project. We call it hybrid assembly, in which for the assembling of the contigs to the chromosome level assembly, we usually do third generation sequencing technique. But for accuracy, we generate data using the second generation sequencing technique. In the second generation sequencing techniques today, the Illumina sequencing techniques, which is earlier owned by a company called Solexa, is now the means market leader. Almost any type of sequencing we will do using the second generation sequencing technique, it will be Illumina. So we have also published a paper back in 2018, and if anyone is interested more on NGS, they can read this paper. So now coming to the various application of NGS techniques. So there are so many applications like transcriptome sequencing, allele mining, metagenomics. Earlier I talked about single cell genomics, epigenetics, multiple genome sequencing, protein binding domain protection, and always also the molecular marker discovery. All these techniques is possible by using the NGS techniques. Now, the RNA sequencing techniques is a sub part of the NGS technique, and in it we use to sequence the RNA from a particular tissue. So, suppose we want to identify a plant is having resistance for a particular disease, and we want to identify the resistant genes in that plant. Then, what we do, we'll take the plants, and in one treatment, we will inoculate it with sterile water, and a treatment we will inoculate it with the spore of that fungus, and after a certain time point we will isolate the tissue, will study the tissue and isolate the RNA from that, and that RNA contains actually the what is the transcriptome profiling of that tissue at particular after interacting with the infection of that pathogen. So the plants having the resistance will obviously produce more of the resistant gene during the infection, because it induces the expression of resistant gene. The viral fungus when spore germinate and penetrate to the plant tissue, plant recognize that the plants have been attacked by a fungus, then it will produces that R gene. So that the copy number of the number of mRNA in that RNA sample will be higher. So by comparing with control as well as treatment, we will identify some of the genes are getting overexpressed after infection with that particular pathogen. So by analyzing and comparing these two samples for transcriptome, we will able to identify the particular gene responsible for that pathogen. Of course, we will further characterize gene through proteomics or through other functional genomics techniques. And so this is nutshell. In nutshell, it is about what is transcriptome sequencing. But just sequencing the RNA is not a single technique. By using multiple iterations of that, we can have multiple information. Like it is also used in evolutionary studies. It is also used in SNP discovery, comparative genomics, even allele mining, gene annotation, and ESTSR mining. There are a lot of techniques are available. We can, by using a single technique and with different combinations, we can have much more information about the genome and about the function of a particular set of genes in a plant system. So now coming to the single cell sequencing techniques. Earlier we are unable to identify a small amount of, very least amount of DNA, but now we are unable by using the third generation sequencing techniques, we can identify a single nucleotide even. So in that we in the figure A, you can see that a particular tissue is also have a certain different types of cells. So each cells have different transcriptome. So if we identify to map what is happening in a single cell, presently we are studying on a tissue level. We isolate the DNA from leaves. So leaves have different kinds of tissue in there. Epidermis there, palisades are there, guard cells are there. So each cell has their different functions. So, but we are studying the cumulative transcriptome or cumulative expression of genes in a particular tissue, but by identifying a single expression of different gene expression in a single cell, we are actually having more resolution. So it is comparable to the Google Map. Just like in Google Map, if you zoom and see the map of a particular our country, you can have you should know that Delhi is there. But after zooming, then you will find the Delhi is there. And after more zooming, you can find, okay, there is a Pusa. And after more zooming, you can find where is an IPV is there. Like that. In particular cell, a lot of things are molecular mechanisms are going on. But by enabling our technique, how deep we can understand, we can identify a new horizon, and what is we can learn about what is actually happening in a particular cell. So by using the single cell sequencing techniques, we can understand new kind of mechanism for response to a plant for a particular stress, either biotic or abiotic. Now coming to the gene editing tools. So gene editing tools usually nowadays gene editing is synonymous to CRISPR-Cas, but it is not like that. Gene editing is basically a tool to edit a gene of our own desire. Earlier, by either using mutagenesis, it's a random, we can induce the mutation, we can induce the frequency of mutation, but where we want to a mutation, that is not in our control. Even in transgenic, we can improve the efficiency of transgenic, but where the tDNA will insert in the genome, it is not in under our control. But gene editing tool, or the first generation, is of gene finger nucleases. In that, we have the certain sets of amino acids that can actually bind with a three pair of nucleotide. So by means calculating the particular DNA sequences, we then have to generate artificial nucleases, and that nuclease will identify that and the sequences and cut the DNA. So the efficiency is very less of that, but in principle we can do that. So by because it is very important, if we are unable to make a mutation in our desired location, then the crop improvement will be very fast, because we suppose it is like that you are throwing a ball to a stump, but earlier it is a random luck, but now you can precisely throw the ball to the stump. So it is like that only. So now coming to the with improvement, a newer technique came, it is TALEN. It is called transcription activator like vector nuclease, in which it by uh, besides designing a certain amount of nucleic acid amino acids that bind to their particular nucleotides, in which it uses factors bind the regulatory elements in the DNA sequences. So it is also better than gene nucleus, but it is not possible for every lab to means have all this information and characterize and synthesize the gene, these tolerant factors, and mutate the genes. But some of the reports are there. It is working, but the efficiency is also very low. So almost a decade back, the CRISPR-Cas, it is cluster regulatory interspecific short palindromic repeats. It is a virus. It is a bacterial adaptive immune system actually. There are two ladies which want to study what is the adaptive immunity system in a bacteria. So suppose the bacteria is infected by certain viruses, and then if it survives with that virus, then that virus will not affect it in future, because virus bacteria what will do, the region, this cluster CRISPR region in the bacteria, this enables to the it cut the virus DNA, and that DNA will is absorbed in the genome of that bacteria, and when next time the virus happen, that part of the virus genome which has absorbed by the bacterial genome is actually guiding the nucleases to complementary binding to the virus genome and cutting the virus genome, and thus protecting the bacterial cell. So by using this methodology, what we do, it is like resistant endonucleases, because resistant endonuclease we will find it in nature, but by using these properties, we can do the recombinant DNA technology. Similarly, we found this CRISPR-Cas virus adaptive immunity in bacteria, and then we adopted it with our purpose. So now coming to the CRISPR-Cas, this is comprised of the two different domains. CRISPR is like the guide RNA, all sorts of things are coming to the CRISPR, and Cas9 is actually a nuclease which binds with the gRNA, and wherever gRNA finds its complementarity in the genome, the nucleus protein Cas9 cuts the genome. Okay, so these are the two ladies, Jennifer Doudna and Emmanuel Carpentier. They have identified this technique, and they are also awarded Nobel Prize in 2020 for their groundbreaking discovery. So coming to the how CRISPR-Cas works. So we have a guide RNA with us. Guide RNA actually forms the CRISPR domain and the Cas9 protein. So it is the Cas9 protein is there. You can see in the figure, the gray lobe are there. This is the Cas9 protein. And the yellow pin-like shape is the guide RNA. And the blue DNA is our target genome. So the pink region is called the guide RNA sequences that we design. And the yellow part is the backbone of the guide RNA. So the backbone of the guide RNA will be same, but the pink portion that bind to our DNA, that can be changed. It is a 20 or 21 nucleotide sequences, and that just like primer, we designed the gRNA, that gRNA sequences, and based on the complementarity, this cut will cut in our desired gene. So, and by there are cellular repair mechanisms for DNA cut, like non-homologous end joining as well as homology directed repair. So by these two techniques, the Cas9 cut the DNA, but cell actually wants to repair the DNA. And in that repair time, some of the times the mutations happen. A particular nucleotide may add or delete, or even a new chunk of gene can be added in that sequences, and based upon their size, it can classified into three type of further in three type of techniques: SDN 1, 2, and 3. It is nothing just like side directed nucleus 1 and side directed nucleus 2 and 3. The SDN 1 is a single base nucleotide changes in the DNA after mutation by using CRISPR-Cas. Then SDN2 is when this nucleotide changes is for much more than 2 or 3 nucleotide, but not higher than 5, 6 nucleotide. And the SDN3 is when the alien gene, a foreign gene, will integrate in the cutting site due to the Cas9 protein. So we have also published a review paper on this technique. If anyone interested, can have a look on this technique paper. And the groundbreaking means decision was taken by Government of India on May 17, May 2022, where they have allowed that the SDN1 and SDN2 genome editing techniques will not be considered as transgenic techniques. Why it is much more means very good news for all the biotechnologies and crop improvement personnel, because when we are going for transgenic, since it has the gene which is never been incorporated in the genome of that particular plant, it's maybe from bacteria or viruses or some other plant. So we don't know how it will react, what is the long term of this transgenic plant. So a lot of different kind of studies and regulatory frameworks are there. So the means developing the transgenic plant for a particular trait is not challenging for the scientists, but getting that plant to the for ready for the regulation from our government or different regulatory agencies is very much big havoc. So by exempting the SDN1 and SDN2 technique from this transgenic regulation network actually helps means remove a lot of means hurdles from the crop improvement perspective, since because there is no transgene is there, so everything is naturally mutation is happening. So therefore it is very much important and will enable us to fasten the crop improvement program across the country. So this is a case study. This paper was published by Stanick et al. In 2021, where they have chosen a Brassica napus plant, which is also called canola. And in wild plant, you can see the taller plant. They have directed the D14 gene, which is struggle estrase gene responsible for the branching pattern in the plant. So what happened? The gene by using the CRISPR-Cas, the gene has been silenced. So you can see that the small d14 mutants are there, in which plant height is reduced, number of branching and number of flowers per plant is improved. So it is helping, because we all know that the taller plants are not desirable because they tend to lodge, and one more problem is there: a lot of nutrients are going to the vegetative growth. So if plants is short, the lodging problem will not be there, as well as the swing source relationship will be higher, and harvesting days will improve, thus increasing the yield. So by using this kind of technique, this is just an example. A lot of different genes can be taken, edited using the gene editing tools, and especially those genes which are negative regulator of desirable trait. That gene can be particularly targeted by using CRISPR-Cas techniques to develop a improved plant. So here is an example in which Japanese scientists, this is a 2022, Japan has released first genetic improved tomato for commercial application. So it is not a theoretical technique. This technique is actually seeing the market and very much important. Even in last year, our Indian scientists at NIPGR have developed low glucosinate line of Indian mustard by using CRISPR-Cas technique, in which they have mutated the promoter, which is responsible for storing the glucosinate in the mustard seed. So mustard plants have a normal glucosinate level, but mustard seeds don't have the glucosinate, which is the economic product of that plant. So these kind of studies will uh, benefit us very much. Now coming to the marker research selection. So we all very much familiar, because it's a very popular technique, and now many of the labs and SUs are and ICL institutes are working on this technique. So here in pictorial diagram, you can see that the mapping population is developed, and like parent 1 and 2 crossed in F1 by using single seed descent method, we will develop used to develop a real population. Either some of the other mapping populations are available, such as near isogenic line, double haploid backcross population, nested association mapping population, and magic population. So these are the population, and besides these population, we can also use available set of germplasm or varieties or land races, and through genome-wide association studies to identify a particular trait and their link marker. So in it, we use a molecular marker to tag a particular trait. Suppose we find that a particular SSR or SNP is responsible for having the high linkage with a particular desired trait, that marker can be assessed for means screening the um, desired plant even early stages, so that we'll focus on that plant only, and that population that don't have the required or desired gene can be discarded, and we will have the desired plant and early stages, as well as we can make the crosses, and some of the times it become very critical. I think that I have also presented this slide. This slide we can be skipped. So now coming to the markers selection. So there are various applications, like in disease resistance, we can use it. We can use it for abiotic assist, tolerance, yield improvement, as well as nutritional enhancement. In mustard, in our case, we are using molecular markers for lowering two antinuclear compound, like erucic acid, which is responsible, is a 20 carbon fatty acid found in the mustard oil, and the glucosinate compounds, which are sulfur containing organic compounds, signatory metabolites that are present in the mustard seed meal. We are using these molecular markers to reduce this two antinutritional compounds, and thus developing biofortified varieties in mustard. So there are also some advantage of mass listed here. You can read here. Now coming to the precision breeding. So by using all these techniques is fine, but integrating of all these techniques is important for getting the optimum yield. Like by using genomic selection, we can identify exactly plants having high breeding values for a desirable traits, and then high throughput high throughput phenotyping. So like high throughput sequencing, the high throughput phenotyping, because manually we cannot take data for all the plants all the time. But by using multi-spectrum thermal imaging cameras, by using like in IRI, there is phytoton facilities are there, so it is enables us to take a lot of data of a plant. So by proper visualization, we can't see the plant is getting stressed. By using hyper spectral images, we can see that which plant is getting stressed. So we will identify the plant that have don't showing the means a stressed situation upon a particular abiotic stresses. And then precision breeding is nothing by it is using these two techniques, the genomic selection or marker data or the our sequencing data, and comparing it with the high throughput phenotypic data to get identifying the what is the genomic location that is governing the particular trait, either it is a gene or QTL, that can be further incorporated into a desired our elite germplasm breeding lines and varieties, and thus developing high yielding varieties having multiple disease resistance or biotic and abiotic stress resistance along with their higher nutrition values. So as I also talked about epigenetic changes, epigenetic is nothing. It is due to the change due to the environment, and there are a few techniques responsible for this. By using this, we can know the epigenetic changes in a particular genome, like so epigenetic crop input enables us the regulation of gene for enhanced stress tolerance, disease resistance, yield, nutrition quality, and hybrid birth, offering a sustainable approach to boosting crop performance because it is highly driven by the environment, and we all know by seeing the changing climatic condition now the environmental influence is becoming very much important. So we have to focus on these kinds of techniques also for developing a improved crop varieties. So one more technique is there, speed breeding technique. I am very short. I will discuss this. Like it is accelerated breeding. By accelerated breeding, we can reduce the crop cycle and develop multiple generations of crop in a single year. So how it is beneficial to us? Because we all know that for breeders, multiple generations are required by for getting a particular desired phenotype or making the particular phenotype stable in the line. We need to go for several rounds of selfing and the pure line selection is there. So many years is required for a particular variety to develop. But by using this speed breeding technique, we can theoretically we can go up to 4, 5, or even 6 generations. We can generate, and what it uses artificial lights. You can see in the picture, it is purple light. It is nothing but blue and red spectra that are majorly absorbed by the plant. So plants are giving the lights that is responsible for getting most optimum level of photosynthesis in the plant. And by using the method tweaking the temperature and humidity, we will get the plants to get the plant to stimulus to produce flowering to grow into the their one is the vegetative and was the fruiting zone. So we want to induce the plant to go into the fruiting zone earlier in their life cycle, so that instead of going taking 140 days in mustard life, instead of going for 140 days, even up to 60 or 80 days, we can have the crops. So by breaking the seed dormancy, we can have 3 crops in a season. So like within 1 or 2 years, we can develop a mapping population, or with the transgenic, or with the even in gene edited crops, we can have multiple generation within a year. So the final product for active trial will be instead of it will taking 8 to 10 years, within 2, 3 years we have the product ready for the testing in the active system. So this is very much important in seed breeding, and now ICR and our government of India is spending a lot of money and manpower to develop these facilities in our country. So with this, I think last lecture I have discussed this artificial intelligence and data-driven decision making, because when the data, a lot of sequencing techniques are there, a lot of genomic data is there, a lot of phenomic data is there, and sensor-based data is also available, and multiple omics techniques are available. So these data cannot be handled, we cannot handle manually. So we need machine learning or AI to analyze these data and develop the desired algorithm and found and reaching us to a particular decision. So which kind of plant and which kind of marker with which kind of agronomic practice. Because earlier we thought about phenotype is genotype G into interaction, any genotype into environmental interaction. These factors are governing the phenotype. But now it is G into E into M is also there. M is management, because a particular variety, even a particular environment, but with poor management or management not given untimely management, it not perform its optimal potential. So with all these to perform very well, to get the maximum yield per unit area with minimum inputs, we have to go utilize these kind of newer techniques. And now computing power is also very frequently available. It's a processor and computers are now very cheap, so we can have develop this kind of models and analyze our data, multi-dimensional data, to get the desired results. So in conclusion, modern micro techniques incorporates NGS-based techniques, omics techniques, gene editing, precision breeding, as well as AI-driven approaches. And the potential outcome from all these will be climate resilient crops that perform much more stable, having the much more stability towards changing temperatures, as well as means water stress situations and all those things. And we have steady and means proper crop productivity even with these fluctuating situation. And we also have the nutritional quality of brassica as well as other crops by using these techniques. And all these things should be in sustainable manner. We should not think about short-term goals. We should also have our eyes on long-term goals to utilize means get the optimum yield advantages by preserving the environment, not exploiting it. With this, I would like to thank you all for patience hearing. Thank you so much.